Okay, welcome everybody to the IRAM meeting series. Happy that most of you were able to get Google Meet going. Hopefully this will serve us well for us meeting together and taking notes and recording meetings. So we'll see, and we can always pivot to a different service if it doesn't work out for us. If anyone knows of someone who wasn't able to get the Google Groups to work, or didn't get this meeting invite, please just let me know or let Chloe know, and we can make sure that they're added. If not, through the Google Groups, we can add directly to the invite list. So a few updates before we get to our speaker today. One thing I wanted to bring up is some questions around interest on forming subgroups within the IRAM working group. So just to mention, for instance, there's the broader AAPS integrated summary of immunogenicity harmonization group that got kicked off. That's being led by a few folks, which will put their contact information in the meeting notes. But there are some other working groups that are starting to come together. To give you a heads up on it, although it's not ready to officially launch yet, the Immunogenicity Database Collaborative is hopefully going to publish soon, and there's already some groups forming around how to expand that data set further. And so some subgroups are forming around how to aggregate and put in preclinical data into the database. Another subgroup is looking to expand the clinical data that's being captured, such as adding pharmacokinetics data and other data points. And then another group who is coming together to hopefully do some automated data scraping of clinicaltrials.gov and maybe the EMA website in order to be able to pull in more clinical data on a broader set of therapeutics for the data set. So there's ideation around those subgroups going on right now, and then once that's solidified, announcements will go out in AAPS, and we'll mention them here to try to see who wants to get involved with those. So,
Dan,
yeah.
sorry, I have a question. What about the maps? We have a maps, AAPS maps working group.
Do we?
It's led by Joachim.
Okay.
It's emerging. Okay.
Yeah, that would be great. So Joachim would be the person to contact for that?
Yeah. You know how I'm collecting infos from different, consortium initiatives, whatever. So you can send me a slide. It's very brief because it's just the beginning and it's an AAPS maps group. Yeah, I've got the ISI harmonization. I have the ADC, and I'm going to add that. If that's okay with you. That's going to be an AAPS working group. All right.
Sophie, do you know what the objective or deliverable of that MAPS working group is going to be?
You know what? Let me check if I've got anything that is of substance. Sorry, putting the wrong one. Bear with me. Okay, what do we have? If you have other announcement, but you can say but what I'm looking for that.
Yeah, so I guess the broader question for the group here, hearing some of those highlights of those groups that are coming up and the deliverables and objectives, wanting to see if there's any interest here, if anybody wants to mention some ideas around other subgroups that could come out to develop, anything around guidance, startup collaborations, work on drafting a publication. So for instance, I did hear some people asking about, there's the integrated summary of immunogenicity group that's coming together to try to harmonize that, but that's really going to be focused on, clinical aspects of immunogenicity measures. The question would be, would there be an interest maybe on coming up with a IRA subgroup who's going to work on standardizing recommendations around preclinical immunogenicity measures and what should go into a regulatory filing, from the start. So yeah, open for that idea or any other ideas that people want to bring up.
So there is an IQ that has started, with the immunogenicity risk assessment group. That's an IQ, and then there is an IQ on, where is the other one? Sorry, I'm mixing up everything. There is an IQ ISI. No, it's the AAPS ISI harmonization, the template.
Right, yeah. I think it's through AAPS.
Sorry. But the immunogenicity risk assessment, that's also AAPS?
Maybe it might be interesting to say here that there are 3 publications on the way for guidelines for harmonization from the EIP. So both, for MAPS, for BC maturation assay, and for T-cell assays. So they're all currently being submitted. One already reviewed at Frontiers.
Awesome. So Chloe, you said T-cell assays, MAPS assays, and what was the third one?
The DC assay, DC maturation assay.
Okay. Awesome. Yeah, so definitely probably not worth it to do any overlapping of those, especially till we see what the publications look like and if there's any particular aspects that might be of interest to build upon from that.
Mm-hmm.
Okay, can leave it there. If anybody has any interest or wants to propose something, we can put it in the meeting minutes, and definitely can talk about it during the next call if we want to start identifying people who might want to get involved in defining what that process might look like. The other thing I wanted to bring up was something that actually Sophie, who's speaking today, brought up. I posted it in the AAPS TPI community as a discussion point, got a few replies, but interested in feedback from this group. So the question was, overall, there's not really a clear understanding broadly across the industry of how many clinically evaluated biologics end up failing primarily because of immunogenicity liabilities. And so we're wondering if there's a way to potentially gather that information in a de-identified way, so that we could better understand how big of a challenge this actually is. Because often those things that fail in early phase clinical trials might not get published on or reported on. So got a few pieces of feedback in the APS community that this might be really difficult to track down internally. Even if they were able to share the information, it might not always be clear exactly why a molecule failed. And there might be, multiple contributing reasons, both immunogenicity and non-immunogenicity related. But yeah, interested in other people's feedback if they feel like this might be a feasible thing for us to do as a community, to gather that information to better understand how much of a challenge this is or how little of a challenge it is. So I'll leave it at that and see if anybody has any feedback.
Yeah, maybe to add to this, the idea was that there might be still some resistance in some companies to do some de-risking ahead of time, like de-risking before we reach the clinic. And we thought that maybe coming up with numbers, like, well, you know, now that we spoke with a lot of people across the industry, on average, each company experiences X number of failures related to immunogenicity. So it's not a rare event that we have to stop programs. So it's helping us get even more data to get more funding, more attention. That was the idea, and really just trying to just get a number per company to start with, right? But I totally understand that for some programs, sometimes it's just not immunogenicity, but maybe for the ones that for which immunogenicity played a big part, we could count these. It's super taboo, apparently, right? Unless you've published it and you've become famous. Cizumab, not to mention it. But I think a number would help, and so that presentation I'm going to give at the CHI, mentioning all these collaborative efforts. I wanted to have that number to start the talk. Say, look, this is where we are with immunogenicity. That's why we're joining forces. I guess within a month, I don't think we're going to have that. Maybe we can have, the ones who can share, they can just give a number. We need to find a way to have that anonymized.
Yeah, it's quite hard. We're actually trying that at the moment internally. And as you mentioned, it's pretty hard to even get to the point to understand if the molecule failed because of immunogenicity or some other factors in the clinic. So even pulling out this data from our internal database.
Yeah.
Easy.
Yeah.
So we keep track of the newer programs, but for the older ones that were already in the clinic, yeah, a bit different.
So I don't know what we could do is maybe send one slider to this group, then people can take that to their management and try and get some like feasibility info. I explain what we're looking for. Would that help? Push. Okay. I can take that action.
Yeah, and we could send it out to this group, and then we could also post it in the TPI community.
Yep.
Great. Awesome. So last thing before we get to today's talk, I just kind of want to put things into context. So we've got a really nice lineup of topics today and then for the rest of the year. And so today we have Sophie Tourdot, who is going to be discussing clinical impact of ADAs, and she's going to be using Adalimumab as an example. And so we want to have lots of discussion around how we think about that, how we think about if immunogenicity will happen, if it will be an impact. And that ties in nicely with our next meeting on October 14th with Morten Nielsen and Joachim Clifford from DTU, the Denmark Technical University. And they're going to be discussing the state of B-cell epitope prediction. So after we think about if ADAs are important, we can then think about what does the state of the field look like for how we could predict whether there will be B-cell epitopes on our molecules and where they might be. And then that'll get rounded out on November 11th with David Preota from MSD in Czech Republic discussing humanist assessment. So we'll look at B-cell epitopes very broadly without the context of whether something is human or not, and then bring that into context of whether a molecule has any human or non-human sequence within it and how challenging that can be from an antibody perspective. So hopefully that'll give us some good context for us here. We're still looking to fill in our December meeting. So if anybody has ideas for that topic or would like to present themselves, please reach out. happy to put you on the agenda. And with that, I will turn things over to Sophie.
Do you want to start with the your introduction slides?
Sure. Do you have those in your presentation?
No,
Oh, yeah.
I didn't add them. Sorry.
Okay, let's see.
Let's see if I can grab them. Trying at the same time as you. Not the best at finding things today. Thank you. Great.
Let's see. Can everybody see that?
Mhm.
Okay. Yeah, so to kick off the conversation, just kind of want to start with a few pieces of information that I know will be obvious to everyone, but it's good to kind of illustrate and put into context what Sophie's going to be presenting on. And so we worked together on putting these slides together so that we can have a robust conversation. So also please, anyone who's interested in contributing, please contribute your thoughts. So, we all care about immunogenicity ultimately because of the formation of these anti-drug antibodies, at least in the context of these soluble biologics. And we care about them obviously because they can impact the efficacy of our drugs and the safety of our drugs. And, ultimately, this clinical impact is at the heart of what we're trying to predict or understand the risk of for all of our preclinical risk assessment tasks, and then mitigation tasks if we think there might be some sort of liability. So the broader question, right, is does this unwanted immunogenicity even matter, right? How bad do these ADAs impact? And if they matter, how frequently do they matter overall? What are the circumstances in which they have an impact? And what are those consequences? And how can we better understand those? So to give an example of how some of these ADAs can impact efficacy and safety, this is one example of pegloticase or Krystexxa, where there are clear indications of how the development of ADAs impacts safety from a hypersensitivity standpoint. Here you get your classic FDA black box warning because of the relatively rare but severe reactions for anaphylaxis or infusion reactions against the biologic. And on the right-hand side is from a publication from patients treated with pegloticase, where it really shows a direct relationship of how the ADAs are impacting drug clearance and efficacy of the drug. So on the left-hand side here, you can see there's an example where you have on the top 2 patients who were responders against pegloticase, and on the bottom 2 patients who were non-responders. And you can see they're reporting on the y-axis here the uric acid concentration in the serum, and this is the direct efficacy readout for treatment for pegloticase in gout. And you can see with the responders, after you get the first dose, you can see the uric acid drives down and it can maintain this low level of uric acid over multiple courses of administration. And that's in contrast to the non-responders on the bottom, where you can see initially you get a drop in the uric acid, but that later very quickly recovers back up and you no longer have efficacy despite the fact that you continue to dose the patients. And I've highlighted this region from 3 weeks to 9 weeks to show you on the right-hand side of where you start seeing the emergence of these ADAs. And so you can see around this same time period, here it's showing this responder on the top versus a non-responder, and same thing for another example on the bottom. And you can see for the non-responder in the dotted line, you see this emergence of ADA titers, which is shown on the y-axis, around that exact same time where you're starting to lose the efficacy in terms of uric acid reduction. And the same thing with the other non-responder on the bottom. Another good example of this was a larger kind of meta-analysis showing how if you look overall for an example anti-TNF alpha antibody, as well as a few other biologics, if you look at the percentage of responders in two buckets, those patients that were negative for ADAs or those patients who were positive, those negative showing in the dark green and those who were positive in the lighter kind of teal. You can clearly see that there's an impact on the frequency of patients that are going to respond to your drug based off of whether they have ADAs or not. And this is illustrated on the right-hand side for adalimumab and infliximab in terms of the clearance of the drug, where if you were positive for the ADAs, you can see a much lower drug concentration post-dose in comparison to those who are negative. And so there's this broader question of how common are ADAs, and what are some of the average reported frequency of those ADAs. This is giving you a snapshot of the current Immunogenicity database collaborative data set, which covers 113 molecules, mainly monoclonal antibodies, but a couple of other modalities sprinkled in. And obviously, if we look at this as a snapshot, it obviously isn't a totally unbiased sampling of biologics. But if you look at it as a snapshot, you can clearly see that there's a wide range of ADAs across molecules, right? So it's not the case that every biologic will have ADAs, and definitely for some, you can even get 100% of patients developing ADAs. So, and the question is, even with this diversity, which context do these ADAs actually matter, right? Because just because you have high ADAs doesn't mean you're actually going to result in clinical failure or actually impact efficacy or safety. And so a few analyses that have been done in the past, a few publications. One is from Vermeulen et al. I think this was back in 2014 or 2015. And so they did an analysis across 51 trials in which ADAs were detected. And so out of those 51 trials, only a smaller percentage of those 51 trials reported on aspects like impacts on pharmacokinetics or efficacy or toxicity. But for those that reported on pharmacokinetics, they found that there was about an equal mix of those that reported having an impact on pharmacokinetics. So if there were ADAs detected, 47% had an impact, reported impact on PK, and about 57% had no impact. And it's a similar story for efficacy, where again, it was about 50% of the time for those that actually reported on impacts on efficacy, about an equal mix of whether there was an impact on efficacy or not. Toxicity wasn't as clear for what they reported in this paper, but essentially overall, only about 20% of the trials reported an impact based or an association of ADAs with toxicity. And so this was from their analysis from the literature, but then they looked at 26 EMA and FDA approved drugs. And across those, you could see something relatively similar. So out of the 26 that they evaluated, about 92% of those had some sort of detected ADAs, so reporting on ADAs. Of that, only 29% had greater than 10% ADA frequency. So 10% is a relatively arbitrary number, but it just gives you a context of, the majority of these had very low ADA rates, but 29% of them had greater than 10%. 42% impact, 42% assessed whether there was an impact on PK, and a minority of those described an impact on PK, so only 20%. In terms of impacts on efficacy, 50% reported on it, and 0% mentioned any impact on ADA rates, and 71% reported on toxicity, and 18% reported having an impact. So a little bit different from their sampling of 51 trials, but you can see, again, there's kind of varying amounts whether these ADAs will have an impact or not. And for the sake of time, I think I'll skip over this. It'll be in the meeting minutes and the recording, but this is another interesting meta-analysis evaluating, how often people are reporting ADA incidents, how often are they looking at neutralization, impact on PK efficacy. And for those that report on these metrics, how often are they seeing an impact on, clearance of the drug or impacting efficacy? So with that, I will turn it over to Sophie.
All right, thank you. Well, let me. Sorry. Okay. Let me try again. All right. Okay, so thank you for the nice introduction on the topic, Dan. So the question here that we looked at was do we have an association between the ADA and immunologically related adverse events? Oh, I got a question from, oh no, it's a question to Dan. So the idea here is that we always look at this association where we said as you said, and one of the consequences of unwanted immunogenicity could be consequences of safety, and this include these adverse events. And so just to talk a little bit about the, it's not, sorry, why is not moving here? So when I put full, it doesn't move.
It's not in presenter mode yet.
Yeah, it was, and then when I move it, can you see presenter mode now?
Still shows the PowerPoint.
That's so strange. Okay. So you know what I'm gonna do? I'm gonna make that a lot bigger. How about that? Better.
Yeah, looks good.
Okay, so just a few terms and definitions, because sometimes we get a bit confused. We confused between the IRS, HRS, or SHRs, and all of that. So infusion reaction for the FDA, and then non-allergic injection site and infusion reaction. Oh yeah, there is also infusion reaction and injection site reaction, all of these. So these are the definitions for this one. Oh yeah, okay, I can't have a rule. And anaphylaxis or hyperacute acute reactions for EMA, which would be the hypersensitivity type 1 reactions, and the non-acute reactions or delayed reactions, depending on what agency is describing them, and it's hypersensitivity type 3, type 4. So type 1, IgE mediated, and type 2, we don't talk about it in this context. Type 3, immune complex mediated, and then the type 4 is the T cell mediated delayed reaction. So today I'm using ISR for injection site reaction, and then HSR for hypersensitivity reaction, and then all of them together, the immunologically related adverse event. Then I would call them the IRAE. So the study, the data that we use to conduct this analysis of the possible association, the data come from a clinical trial, which was a multiple switching between the biosimilar adalimumab and the reference adalimumab in patients with RA. So I thought I'd give a little bit of details on the biosimilar clinical trials, because sometimes that's something that we're all familiar with. So for the biosimilar, you've got different steps. You've got comparability that shows that it's biosimilar. Oh, and Daniela, interrupt me if I say something that's not entirely correct, please. And then there is the switch, meaning that you can switch for the biosimilar. And then there is interchangeability, which is the next step. And this, the impact is more for the patient, in the sense that if you have demonstrated interchangeability, then in that case, the switch between the biosimilar and the reference product can be made by the pharmacist. There is no need to go back to the prescriber and ask the doctor if it's okay, which is the case if you have switch only. So this is another level, and really the interest is to the patient. So there is more flexibility. Thank you, Daniela. So now, how do you demonstrate interchangeability? Then what you have to do is you have to show, it must be a biosimilar, and then you have to show that they have the same clinical results. And you must show that there are no additional risks when you have the switch back and forth with the reference. So it's no safety risk and no impact on efficacy. So from the guidelines, you can have an idea of what type of protocol you should design. So an important point here is that when you start looking at this switch, the difference might be subtle. So it is recommended to use PK or PD as endpoints, and they're more likely to show differences compared to efficacy endpoints. And to really look into the impact on PK, there is a period that is called intensive PK sampling. So basically, the protocol, you start with your regular administration, so that's the leading period, and then you start a switching period. So you're going to have an arm that doesn't switch, and then you're going to have an arm with switching, and you have to do 3 switches. And then at the end of that period of switching, the various length, you've got this PK endpoint sampling. And what it means that you're going to have many sampling for PK measurements during that period. So you have a closer, more granular idea of what's going on with the PK. So this was the design of our trial. So we've got this 10 weeks lead-in period, and then we have a randomization, and we go switching arm or non-switching arm. So Abrilada is our biosimilar, and so you've got, you see the switch between the two, Humira, and so different per treatments, and during this period 4, we have that sampling. So it's week 30 to 32. So here's the detail. So the last treatment, so the last injection of the biosimilar is at week 30. And then we start the sampling for PK. So these are the days after the last dose. So this is going to be between week 30 and week 32. Week 32 being the last week of the trial, the last dose. So this is the paper with the results. And I'm going to show graphs from this paper and graphs from the original paper that described the interchangeability study, because we've got a similar graph, but we do have the ADA and NABs on top of it in terms of analysis. So a few notes: since we had demonstrated interchangeability to conduct the analysis, we could pull the data. So it's Adalimumab altogether. It's Adalimumab, which includes the biosimilar and the reference product. That's what we used. And then for the PK intense sampling period, so we have the PK, and what we use as ADA measurement is the ADA measurement at week 30. So it was the ADA measurement at the last treatment before these 2 weeks. The safety population, it's everybody that were included, and I had at least 1 dose of either. So the PK over the course of the study. So this is looking at the 2 arms, and so you can see when the PK was measured, different days, and then roughly the PK evolves the same way in the non-switching and the switching arms. And so that was the original description. And then the ADA measurement, this was what was observed in terms of ADA and NAB frequency. So it's at the end of the leading period. So that's week 10 when we randomize, and at the end of week 32, that's the end of the study, and that includes that period of sampling. So if you look at the ADA and NAB frequency, at week 10, when we start the study, the interchangeability study, ADA positive patients and non-switching arms, so 26, 28, that's very similar. In terms of NAB, that's very similar too. And at week 32, then we have, again, something very similar, 47, 49%, and then 8% for NAB for the 2. So when we looked into the detail, then that's this table here. So we have, at the end of the lead-in period, we have 427 participants, and we have 250 participants that are ADA positive, 61% of participants that are ADA NAB positive, sorry. And then we calculated the percentage of the persistent ADA and persistent NAB. So then we have 51% of the patients that are persistent ADA, and then we have 9.8%. Sorry. I know I'm not the best at describing that table. I'm getting confused myself. Okay, and then 9.8% of patients that were not persistent. So now, if we put together the PK that was measured and then the ADA and that measurement, then we see a different picture, because then in that case, so you have the concentration, the drug concentration here, and then over time during the weeks during the trial, and you can see that there is a huge impact on the ADA status on the exposure, because this will be, I will read Mike your comments. I can't read at the same time. So we have a huge impact on the exposure in ADA positive patients that are shown here versus ADA negative patients here. So that's the impact of ADA. Now, if we look at what happens during that period of intensive PK sampling, this is what happens to if we look just at the mean PK over this time post dose. So concentration decreases, but if we look now in terms of NAbs, so we look at the same period, the same samples, and we compare this PK, we relate this PK to the ADA and NAbs positivity at week 30, then we can see that there's huge impact of the NAbs on the exposure, and the impact is almost blanking. Do you see a huge reduction? A huge reduction when you look at the NAbs. So now, from here, we wanted to see whether. Yeah.
I don't know if
I'm
you
sorry?
I don't know if you specifically looked into this, but is there a difference between patients that are ADA positive and patients that are neutralizing antibody positive in terms of impact on exposure? Like, is there an indication that if you have neutralizing ADAs that they somehow impact your exposure more than just having, being ADA positive?
Oh, right. I see. So this is the impact of the ADA. So the impact is lower than the impact of the NAbs. So that was your question, comparing what the impact of the ADA versus the impact of the NAbs.
Right.
Oh, but then in that period, in that intensive PK sampling period, I can't remember if we did that analysis. I'll have a look. So now, using these studies, we look at the association with the potential immunogenically related adverse events. And so what we had in the study that was very that we pay attention to is that we have. So this is, for instance, the data for the ADA negative at week 10. So what we had when what day the study, the assessment of the ADA was done. I mean, the sampling for assessment, and then when the physicians, the clinicians start looking at the adverse event. So we could see, we could really look at the adverse event after that ADA sampling. Do you know what I mean? I don't know if I'm really clear, but we knew that it was really after, and we had an idea also of the time when it develops right after the measurement. So we see, say for instance, we took the samples here at day 69, and then we started the study at day 85. So that tells you what kind of ISR you would see in that case. So when we looked at the overall results, the conclusion was that within the 4 ADA positive patients at week 10. So again, week 10 is when we randomize. So if we look at the 4 positive patients at week 10, we had 2 patients with 1 occurrence of ISR, 1 patient that had multiple occurrences, and 2 patients with hypersensitive reaction, and one of them is the one with the multiple occurrences. That's why if you do 2 + 2 + 1 is 5, but it's 4 patients, because 1 patient had the one with the hypersensitivity reaction is the one with the multiple occurrence of ISR. Now, the thing is that in the ADA negative patients, then you see ISRs, you see multiple occurrences of ISRs, and there is even one hypersensitivity reaction. So looking at this patient level, the conclusion was that there was no apparent association between the status and the adverse event. Now, you've seen that from the table and the numbers, right? The caveat is that it's a very very small data set. So we could not do stats and we could not see whether, it really was a significant difference. So what we hope is that we encourage sponsors to do this kind of like very similar investigations so that we have more ideas of this, because this is just one study, it is small data set. But looking at this study, we didn't observe an association. So thanks, big thanks to my co-author, because as you know, I am still learning clinical immunogenicity. So really big thank you to them. And thank you for your attention. Do we have any questions? Thank you, guys.
Maybe I'll kick off a question or a thought. Adalimumab is one of these classic examples that I don't know if other people get this from leadership teams, but where they say that, yes, adalimumab has a very high level of ADAs, but from a clinical perspective, it was still a blockbuster drug, right? And still very successful. And so therefore, we don't really have to worry about ADAs as much. And it's also an example of, I guess, clinically where people will just dose over the ADAs, right? If you get ADAs and reduced exposure, they'll just add and increase the amount of drug that they're giving the patient. So I guess, yeah, how do we think about that here, right? Clearly, it's you're getting high incidence of ADAs. It's clearly having an impact on pharmacokinetics. You're getting neutralizing ADAs. But practically speaking, I guess since there isn't an association with any adverse events, you can just dose over it. So I guess, yeah, how do we interpret this? Is this a common thing with monoclonal antibodies that you can just dose over it? Is it common that you don't have an association with adverse events?
I think the adalimumab case, going back to just the beginning of your comment, is the one that, yeah, it deserves us internally when we really want to push for de-risking and pay attention to immunogenicity, because then we get, oh well, but look at adalimumab, you've got ADAs, but the impact, doesn't the impact of the ADA doesn't prevent the drug from being efficacious. And so always go back and say, well, yeah, in that case, it is the case for this, but you don't know with a new drug that you're gonna bring to the clinic, you might have ADAs, and that's the tricky part, right? That's we also want to discuss is, yeah, are there any indications for us that you're gonna have consequences? How do you determine this? What are you folks doing in your companies to get a sense of that possible impact? I mean, on safety, we have, when we do the immunogenicity risk assessment, we do have the safety component that sometimes is, it's obvious in terms of severe consequences, and then usually we put that there is a risk of ISRs, there is a risk of HSR, but do we only learn in the clinic?
Yes. Danielle, you got your hand up. Do you have any thoughts on this? Danielle, we can't hear you.
I can't see the hands. Oh, Daniela. We can't hear you.
Nothing yet.
Shall we go to a mic in the meantime?
Okay. Yeah, so for Adalimumab, I may have missed it, but did your patients get any co-medication? But because what we see is that with the okay, because
Yeah.
Now you can hear me?
Yeah, that you.
All right.
Yes,
Sorry.
methotrexate.
And with that,
So,
you can
yeah.
see a big difference also in the development of ADAs.
Yep.
Daniela?
Yeah, can you hear me now?
Yes.
Oh, wonderful. So two things that I want to mention. One is that the questions that Sophie's asking is one that we ask ourselves a lot right now at the agency. We're really thinking very deep and hard about it. And sort of together with that question is also, to what degree are the switching studies informative, particularly when they're so small. So I just wanted to make sure that everybody understood that, particularly for monoclonal antibodies, you will not always be asked for a switching study. And at this point, if you have a good risk assessment, you might want to discuss with the agency before you even plan a switching study. So I wanted to make sure that nobody came out of the meeting thinking, oh, we have to do a switching study, because it is very possible that you won't have to, just because we are accumulating, we're learning from what we see, and we're trying to evolve as we see. But this is, I think that Sophie really raises a critical question, right? To what degree, to what degree is an immune response really an adverse response, and how can we really understand that in terms, long term, right? So you have an immune response, you have a very short switching study, you may not see anything there, but will you see something as you expose that patient for a few years? We really need to accumulate a lot more data. So I'm happy that Sophie and others are trying to really collect more clinical data so that we understand, and we don't sort of make assumptions of no impact on clinical safety when there could really be an important impact. So just that.
Sophie, maybe a broad question for other people. When you detect neutralizing antibodies, how often does that correlate with impact with efficacy?
Ooh.
Is it like as expected correlation? Is it surprisingly like low correlation?
I don't, top of my head like this, I can't give you an answer, but I think in one of the slides that you had, in one of these, in one of the meta-analysis, there might be the answer somewhere.
Mm-hmm.
Or is it something that we can look at using the IDC?
Yeah, unfortunately, the IDC.
We don't have efficacy yet.
We can look. It does mention whether it reported any impact on efficacy, but that was pretty rare that people actually said something about efficacy, whether it was impacted or not impacted.
And even I was looking at the impact on PK, looking through quite a few labels. Then a lot of the time it says that the impact on PK is unknown because there were not enough data points to report the impact on PK. So that's another thing as well, the impact on PK. If you look at the label, because that's all you said, that's all you have access to, you won't necessarily be able to see that there, because it wasn't say reportable.
Mhm.
Regarding the impact on PK, we use our QSP immunogenicity prediction model, and so we simulate a clinical trial, 1,000 patients, and then we look at the predicted AD incidence. And then we also can look at the impact on PK in terms of exposure. So that's the only tool so far we have. So no answer for safety, but we have a prediction. So we know what, with the caveats and everything, but we can have an idea of the possible impact on PK. And the way we used it was to design the next clinical trial, looking at, okay, if we change the dosing regimen, what happened to the exposure? So what do we need to do in terms of every 2 weeks, every 4 weeks, what dose? And you can input these variables, and then you see what happens to your exposure. But safety.
Mhm. Well, it's interesting from the impact on PK, I guess. Is that just predicting titer and then associating increases in titer of ADA as having more of an impact on PK? Because from a mechanistic perspective, the QSP model doesn't understand, where the antibodies are hitting on the drug, like where the ADAs are targeting in terms of
So
epitope.
yeah, so you're right. Something that the model doesn't do is differentiate between AD and NAbs. That's another component that we'd like to have at some point.
Cool. Yeah, I think there's some great components here that we can bring to the discussion with Morten Nielsen next week in terms of understanding where epitopes and
Exactly.
the impact of those.
Yeah, and so a question I had for the group as well is how much do we know about the development of NAbs? Sometimes it is said that when you have high titers of ADA, you're going to see NAbs, but it's not necessarily always the case. Do we have any criteria? Do we know when we look at the response, any indication that NAbs are going to develop? Is it linked to anything in particular in terms of timing, or is it epitopes only, or? Do you know any reference?
No correlation. We did not see any correlation so far. If you're unlucky in your epitope is in CDR region or whatever, then yeah. Okay, but otherwise.
Yeah, do you have any studies looking really at the B cell repertoire, the evolution of the B cell repertoire? No, on one when we had the Iberis consortium, looking at the B cell repertoire, but it was in the context of B cell repopulation after rituximab treatment. So it wasn't so. That's a different case again, because you've got the B cell depletion, but the same kind of analysis, looking at the B cell repertoire, the antibody repertoire that develops over time, and any?
I think they only looked into the, the B cell receptor piece and not the epitope that was recognized.
Yeah, I was just looking at the.
Yeah, and assays that can really give you the epitope are tricky because, for example, for some of the assays you need a lot of serum, and you are not getting that from your patients. You are getting that maybe from your healthy donors, but that's not in the indication, and even that is pretty tricky. Or what we did for some of our studies, this linear epitope mapping is also pretty limited. As in the name, linear epitopes, and we know that we have a lot of conformational epitopes.
Yeah,
So,
actually, I think that's definitely a question I want to ask Morten at the next meeting. Is what he thinks about this when we look at linear epitopes, because they do exist, but
I think that
there weight? Are there different weight for linear versus structural? Sorry,
there's an old publication that stated that actually the majority of epitopes should be conformational and not linear. So I think it kind of makes sense because if you think about the structure of a molecule, then if you think about a helix, some of the amino acids on the back of the helix are just hidden. So the epitope will be bits and pieces of that helix. Of course, if you have a loop, then you might have a linear epitope actually. So this makes sense, but it also makes sense to me that actually the majority of epitopes should be conformational.
And with this linear epitope mapping, you may be able to catch the major part of such a conformational epitope. But that's luck then. That's gambling in the end.
And I think that in that case also these circular peptides don't really help because in the drug they are not circular. They are either in a helix or in a beta sheet or in an interjunction of the two. So I mean we are also interested in learning what tools might be there that are more suitable in detecting epitopes and confirming epitopes, because I think these are some of the major struggles that we are still facing.
Yeah, but all of the other assays we had a look at have their own challenges. So nothing perfect at the moment available, at least to our knowledge. But I'm also looking very much forward to Morten's talk, and because so far I have also not seen any good B-cell epitope prediction tool. And if there is something new coming up, I would be very interested.
When you say no good tools, that's when did you say try one and you ran the molecules and say molecules for which you did the epitope mapping of the ADAs and then it didn't correlate at all or poorly?
I mean, we did not assess a lot of them with clinical material, so that's a challenge because some of those tools might work perfectly well for a monoclonal antibody binding to something, right? Or a monoclonal antibody binding another monoclonal antibody, but for a polyclonal ADA response, these are typically quite a challenge. I mean, there are a few technologies that are based on LCMS, so these HDX or something like that, where you kind of mark the molecule, the exposed residues, and then you're trying to deduce which regions were not affected by the treatment. And those should be covered by the anti-drug antibody. But of course, if you have a mixture of epitopes on your molecule, and then also you may not have your perfect ratio between drug and anti-drug antibodies, then you might just not see it. So I think for a polyclonal response, it is extremely challenging.
Yeah, and the coverage is also not perfect always for every molecule. So
yes.
Yeah, so the prediction of an epitope binding of a specific, so if your antibody binds to a specific epitope, that's there. That's already there, and there are some tools available that are quite okay. But yeah, with this polyclonal answer.
I wonder, Dan, if we could think of a subgroup doing a B cell kind of retrospective analysis with the prediction and then what's known for the different molecules and looking at if there were NAbs that were induced, or I don't know, just thinking out loud. Would there be room for a B cell subgroup of some sort? So I mean, no T cell, but a B cell, and find what is that we need to explore with the prediction, with the characterization, what we know where the gaps are, what is feasible, in terms of retrospective analysis of the data that's available and using the IDC. Any appetite?
Interesting questions. Like just a general B cell immunogenicity subgroup. Then
yeah, a subgroup with all these questions that we have.
I think the interest might be there. The data is potentially not there.
We started, but we start with what we have, right? So we say, okay, we do prediction for whatever, and then we look at what we have. We look at what we can share, if we can share. We look at what's published. Even say if it was the first goal was to to do like a where are we, what's available, what do we know, and then what is the aspiration goal, what would it take to get there, what we don't have. I don't know, just a thought, since we're discussing B cells and prediction and stuff.
Yeah, so Sophie, I think, were you trying to put that together for EIP, and I guess it just didn't come together?
The B cell group, we haven't put that together, no. So we don't have a B cell working group, that's for sure.
But I think also there the scope was a bit different, because the idea was to work on assay harmonization, and since not many
for that one
companies are running such assays, for candidate ranking like that, there was kind of a bit of a limitation. So I think there are, a lot of B cell experts in the field that are certainly, it makes sense to connect them and to maybe explore some of the questions that you mentioned, Sophie.
I think it might make sense to have an APS group. We for the EIP, NCRA, that's right. We just I had the subgroup. We were 3. So at some point, highly motivated, but very little to discuss. Rapidly, very little to discuss. So we just thought, well, we're gonna put the usual sentence in in the paper. We can write something separately. All right. So I hear that I might have at least 2 people interested. I've got Mike and Annette on board.
And probably Elisabetta as well, our boss. She's
something is coming out of that,
the expert in our area. So
So Dan, how does it work if we come up with a new idea for a working group, a sub team, or how do you call it? How does it work? We just you contact the your community, or how does it work?
I don't think there's any official thing that has to be done. I think you can just put it together and then figure out the objectives amongst the group of people who want to get involved. And at most, you can solicit for more participation from the different AAPS groups like IRAM and TPI and stuff like that. but I don't think there's any formality unless you want to have like a formal section in the AAPS web portal or something like that. But just to put people together and get started with an initiative, I don't think there's anything
Cool.
for that.
Are you game?
I'm always game.
Love it. Chloe, I can hear your enthusiasm too. Christopher, Bruno.
Enthusiastic, yes, but as we mentioned before, from an NCRA perspective, it's not directly always the highest part of expertise, like what happens in the clinic and how to, especially the B cells, it stays a bit an enigma. But enthusiastic, yes, for sure. If I can contribute a lot, I don't know.
Well, do you know if there is data gathering, as you know from experience, the more the merrier.
Yes, that's true.
Okay, then let's try that.
So we'll definitely capture in the notes, and
Yeah.
Sophie, if you want to follow up on it,
yeah,
you'll have people interested. And yeah, I'll try to remember to bring it up during the call next month when we're talking about the topic of B-cell epitope prediction.
I'll try to have like just a one slider copied from other groups and see stuff, that objectives and stuff.
Awesome.
Cool.
So really nice discussion. Obviously went like 15 minutes over, but that's fine. Whoever could stay can stay.
Scientists, right?
Yeah. By the way, Sophie, will you share the slides?
Yeah, absolutely. I can send them to Dan, and then we'll put them wherever you want.
Thank you.
Of course. Thank you, guys.
Thanks, everyone.
Bye.
Bye.
Bye.